we're going to move on now to questions. And as is customarily the case, the questions come from committee members, since these webinars are designed to be information sessions as far as the committee is concerned. And I'm going to turn to Wayne Landis to get us started. Wayne's on the committee and has a question he'd like to ask, and he then is a bit short on time. So Wayne, are you ready?
Yeah, thanks, Jim. And David and George, thanks for an interesting talk. My question is based on some of my research, but also because we're trying to incorporate risk assessment into our evaluations. We found that in our models, in our experience, especially in our models, because we can play with them so much, that models of invasiveness and their impacts are very sensitive to initial conditions. I'm talking from a mathematical point of view, but also when we have data and we're just looking to do the what ifs. Also, we have a lot of features that are inherently probabilistic, like how many probabilities get there, what fraction is going to actually survive, and so forth. I mean, you could write distributions for that. So we find that very small changes create lots of different kinds of outcomes. Also means that you're going to need some precise knowledge in order to make reasonable predictions. Also has problems with extrapolation. Are the data that you've gotten for one particular location going to be useful in making reasonable predictions for other locations? And also in comparing what you expect in the field. Because if your models are showing that if you make a run, you get establishment, make another run, you don't, and it's really just due to the stochasticity of it, how do you design experiments, and how many experiments do you have to do in order to parameterize those aspects of it? So my questions, which are based on that, are two. Does your extensive field experience, both in terrestrial and marine systems, does that really confirm what we see with our modeling efforts, and with some of the stuff much smaller than what you guys have done, like with green crab and so forth? And what kind of experiments, can you imagine experiments that we could do to actually get the information to parameterize the models so we can make reasonable predictions when we go from a small site to a larger scenario? Thank you.
David Lodge, you want to start on that?
Yeah, this is David. Yes, thanks, Wayne, for that observations and the question. First of all, I think your experience, as you described in your model world, that is sensitivity to initial conditions, the need to parameterize multiple probabilistic variables, the requirements for extensive knowledge for each one of those variables, and then still being left with uncertainty about extrapolating from the knowledge from one location to another. All of those are not only apply with great strength in the model universe, as you described, but in my experience apply equally strongly in the empirical world. And this is what makes me so cautious and perhaps provide unsatisfactory comments about risk assessment, because it's conceivable to me that you could gather all that information to better parameterize a model, but we've had several long and reasonably well-funded grants to try to do that sort of thing for different examples, ranging from dispersal of an aquatic plant regionally in the Midwest to global dispersal of organisms by ships. And even with a concerted effort by a multidisciplinary research team, I worry that we've not gotten, in most cases, to the point that it would be very useful for risk assessment of a particular novel gene drive application. So let me just say that another way. The risk assessments that I'm imagining that would result in confidence that I think would satisfy a scientific community or the public would require enormous resources and be quite long-term.
Mm-hmm. Mm-hmm.
Yeah, this is George. George,
go ahead.
I couldn't agree more. Plus, there's so many events that are, I would say, not predictable, that, these one-off kinds of things that, I would say even with the best of parameterization of the initial stages, we might not be able to predict. Yeah. Maybe you could put into a risk, sort of a assessment scheme and include some really rare events. But, many of those rare events are the important ones.
Yeah, I completely agree with that. That's what we see too. And just to follow on, the risk assessment depends on what people need to make a decision. And a properly done risk assessment should be able to give those uncertainties. So not just calculate the risk, but also the uncertainties. And it seems like what you guys are most skeptical about is reducing the uncertainty enough, given what we know now, to make reasonable predictions. And perhaps, and this is kind of a leading question, but important, I think, for everyone to hear, it sounds like what you guys have done are talked about outcomes and the uncertainties. And perhaps the most important part about any risk assessment at this stage is to correctly describe the uncertainties in this process. And at least that's my take, and I kind of like your feedback on that.
This is David Lodge again. Yeah, Wayne, I think that's right. I mean, we've spent a lot of time, again, in a multidisciplinary research project, just to give you one example, in recent years, trying to best describe the likely impact with uncertainty of an invasion in the Great Lakes by Asian carps. What would happen to the food web and what would happen to the economy? Well, we've got answers, and when we've published some papers, and there's another one forthcoming, of course, but the unsatisfying part is when we quantify uncertainty, which I agree is essential and the most useful part of a risk assessment, the uncertainties, even with a lot of effort by a lot of people over a long time, the uncertainties remain enormous.
All right.
George, do you have anything? Oh, sorry.
No, I think that's right. I think, we can identify what areas we need to be worried about. And I think David Lodge's work shows that really well. But, still, we know those uncertainties to be huge.
Okay, great.
Thanks for accommodating
Let me quickly follow up. So one of the things we could do in this report is add in some cases. Do either of you have what you feel is an ideal case of when things were done, quote, right, unquote, in a way to mitigate things, and you think that could be a pretty good model?
Well, this is George Roderick again. This glassy-winged sharpshooter introduction in French Polynesia is not a bad model in that, we worked with the local governments. We did, non-target, assessment beforehand, plots that were replicated and compared, over time, and then, sort of a biosecurity assessment at the end. And I would say, working with the biocontrol people and who now have a set of sort of best practices, this might be a model worth looking at. And, there's a pile of papers now, and I could send you those, plus reviews.
Well, that'd be great. If you could send those along to Audrey, that would be great.
Yeah, and
she can get them out to the committee. Yeah,
and then the work on Tetiaroa by James Russell, who's a rat invasion expert in New Zealand, is an interesting one too. For a vertebrate example. And
okay.
There are, well, your committee already knows there are some mosquito examples ongoing.
Right. Right. Good. Good. Very helpful. Let me go on to another committee member, Jason Delborn, who has a question. Jason.
David and George, thank you for really informative talks. Those were really excellent, and I think you've given us some good reason to think about both invasive species and biocontrol as really good analogies as we think through implications of gene drive. What I'm thinking about is the way that you've described our understanding of invasive species and biocontrol suggests that these uncertainties aren't going away, they might even be growing. And in that sense, you might be suggesting that the practice of biocontrol is or should be slowing down, and yet that's in the face of increasing impacts from invasive species and the need to do something. And so I'm wondering if you could talk a bit about the field of biocontrol as professional practice in terms of how practitioners and scientists who study the practice think about the balance between the uncertainties that we know about or that we can imagine and the need to take action to prevent ecological damage.
So this is George again. Maybe that's more relevant to the terrestrial species, but I would say especially the weed biocontrol people have now a set of established guidelines where they have tried to consider these things. There is some risk assessment involved. There are economic considerations as well. I think that might be a good place to look. Also, as David mentioned, there's a long history of these things, and the research is, or the literature is full of examples of successes and failures, and some of these have been studied in retrospect. But I would say, one of the big failings of biological control programs generally is that there's no money for follow-up studies. So the problem goes away, and then what would really be interesting scientifically would be, well, what happened to the communities? How are these things now being controlled? Are they controlled by the biocontrol agent, or are there other things, or are diseases and other parasitoids and pests or predators important? So I do think the biocontrol literature is a great place to start, especially the weed biocontrol people have thought a lot about risk. And I think they'll also, if you look there, I think you'll also see that what's really lacking are follow-up studies which people could use to actually understand what happened.
Okay, great. Jason, does that do it?
Yeah, just a quick follow-up then. One is, are we getting better? Are we becoming more confident, as we might be entering an age when we have a whole new set of tools to do things like biocontrol? And then related to that, or perhaps a little different, is are there places where you think that the analogy breaks down between thinking about biocontrol and gene drive? And David's talk mentioned, a helpful distinction between gene drive that would be aimed at extirpation or extinction versus changing the species. But are there other aspects of gene drive that make the analogy break down? Thanks.
Well, maybe David can summarize, but I thought he did a pretty good job of pointing out where, there are evolutionary considerations that, we don't typically think of for biological control.
Yeah, this is David, and just to go back a moment, Jason, to your earlier question about biocontrol, I thought George's example and everything George said, I thought was right on. And George is more of an expert in biocontrol certainly than I am, but I would observe that I think biocontrol efforts have probably slowed down in recent decades because they have gotten so much better, and that is more incorporating more rigorous risk assessment approaches beforehand. And so it takes more time, more resources, both to do the risk assessment ahead of time, and as George said, we still fall short with the follow-through to see whether the risk assessment was actually good or not. But I think the ratio of successes to failures, I think, has gotten higher with time, partly because the risk assessment has gotten better, and that one result of that has been fewer introductions for biocontrol. But I do think that's probably one of the most analogous areas to look at with respect to risk assessments for putting a new sort of organism in the environment. With respect to the question you just asked, Jason, it seems to me that one of the most, the applications that certainly I get most excited about in terms of potential benefits is the eradication of invasive species, and where you're not like rats on islands. But even there, I guess what, I felt like the assignment I had was to think about the what-ifs, what could go wrong there. And unfortunately, there's a pretty long list of things that could go wrong.
Okay, very good. Thanks. Is that good, Jason?
Yes. Yes, thank you.
I'm sorry, Jim, can I jump in one more time? I realize sure. I didn't really answer your question. I think, Jason, you asked, is there anything unique? Does the analogy break down? And I think with respect to biocontrol, it's the drive part of the gene drive that makes this unique. I mean, in any dispersal event, in any escape, the probability of the organism that's been modified taking off in a non-target place is higher than with any of the organisms we've traditionally worked with in biocontrol or otherwise.
Yeah. Good point. Great point. So here's a question from a committee member, Paul Turner, from Yale. Paul had to go off to a faculty meeting, which we can all relate to. Here's his question, and this goes to Dr. Lodge. If we had unlimited resources, what could we monitor in natural environments to gauge off-target effects of gene drive releases, especially in terms of community species composition or stability?
Yep, this is David Lodge. In terms of community composition, I think we have more new tools these days, particularly genetic-based tools, to, at least at a crude level, monitor what species are present and which aren't, including the use of environmental DNA that we and others have worked on a lot in recent years. But in terms of identifying indicators, indicator organisms, indicator processes, indicator functions that would feasibly and affordably tell you about a level of unacceptable non-target change, I find it really difficult. And I'm afraid I fall back on saying that's going to be a case-by-case set of decisions based on what you know about the organism, what the goals of the release are, and what are the most likely outcomes that you can imagine. But there's an enormous literature on ecological indicators, and it's enormously contentious, really, because it's difficult to come up with simple, easily and affordably measured things that give you the kind of information you want to know. Wish I had a better answer.
Yeah, yes, I think we probably all wish you had a better answer on that one. It would be great. But the situation being what it is, you're exactly right. We have time probably for one more question from the committee. Is there a committee member out there who would like to ask one more question? All right. Hearing none, I think at this point we're just about out of our time. So let me thank both Dr. David Lodge from Notre Dame and Dr. George Roderick from UC Berkeley for sharing your time with us with these really wonderful presentations. They really are very helpful as we begin to continue our deliberations on the committee.